They're very light.
Hello. Yeah. So good morning, good afternoon, good evening, everyone, depending on where you are joining us from. My name is Oluwasegun, and I'll be the moderator for this webinar. I'm very delighted to welcome you to today's modeling organized by PMXAfrica. In today's webinar, our guest speaker will walk us through practical approach on building PBPK models and how to implement them using relevant software. Before I call on our guest speaker of today, I would like to inform our esteemed participants that we do not want our guest speaker to be interrupted while delivering his presentation. As such, if there are questions, comments, we implore them to drop them in the chat box, which will be addressed during the Q&A session of this webinar. On this note, I would like to call on Babajide, our guest speaker, to please begin his presentation. Hello?
You are very loud and clear. Don't worry.
Thank you
Good.
very much for the introduction. I'm going to share my screen. So, can we see my presentation slide?
Yes, we can.
Okay. So, welcome back, everyone. And again, I am excited to continue this pharmacometric series with you. The last time that we met, we dived deeper into PBPK modeling, and we talked about the workflow and relevant equations that we used. And I hope you were able to review those concepts. And also, if you were not able to review the concepts before now, or you are just joining for the first time today, the recording from those sessions are available and you can have access to them on YouTube. So for our time together today, what I have planned is to first go through like a 30-feet summary of PBPK modeling implementation. This is something that we covered the last time extensively. So what I'm going to do today is just to give you a very brief summary. And then after that, I am going to demonstrate working with PKSIM, which is the software that we are going to be using. It is an open software for developing PBPK models. And I'm going to be showing you or introducing you to the interface. And finally, I am going to be wrapping up with a case study to demonstrate PBPK model building with you, which is where we are going to be spending a lot of our time today. So without further ado, let's get right into it. And I would like to get off camera so that I don't get distracted. Just give me 1 second. Okay. All right, so here is a simplified system of ordinary differential equation that we normally use to describe a whole body PBPK model of a drug. And if you were with us from the start, I started with an hypothetical drug X. So I want to maintain that consistency. So let's say we have this equation for an hypothetical drug X. So basically, when we are building a PBPK model, this is all, this is not all, but this is basically the backbone of what the equations look like, okay? So if you look at these equations, they are ordinary differential equations that have several parameters, as you can see on the slide. So let me take a pointer. So as you can see here, there are several parameters, the dose, the Ka, several of them, okay? So we can bucket all these parameters into two. There are some parameters that are specific to the system, and there are some parameters that are specific to the drug. So let's take, for example, let's look at parameters that are specific to the system. When I say system here, I am referring to the biological system that we are modeling. So in this case, we want to build a model for a human being. So there are parameters that describes the human body. For example, if you take the Q lung as an example, which is basically the blood flow to the lung. The blood flow to the lung is a property that is specific to the system, and the value is averagely constant between any 2 or 3 or 4 individual on the average level. So another example of a system parameter would be the weight of the liver here. So this is also something that is intrinsic to the system. And I said weight, but I represented it as a volume because, you know, to calculate concentration, we need volume. So there's a way to convert from weight of the liver, which is something that is easier to measure, to volume. So then the second parameter that we also have are parameters describing the drug. And example of that would be clearance X here, which means the clearance of the drug X. So this clearance, the value is a value that is specific to a drug. So if I change the drug, I have to change the value of the CLX. It's not going to be the same. If I change the drug, I have to change the value of the absorption rate constant Ka here, because it's not going to be the same. So the same goes. If I keep the drug, but I change the biological system from a human being to a dog, then all those system parameters representing that of a human being has to be changed from being those values for a human being to those of a dog. So it goes like that hand in hand. So today we are focusing on building a PBPK model for a human being. Okay. Now, the last time that we met, we said all these parameters, all this equation, we can put them together and then use it to predict drug concentrations. So how do we do that? I mentioned that one way to do it is to write all the equations for both the system, the drug parameters, the dynamics, which is basically the differential equations. We can write it ourselves from the first principle. And that is when I say we use different ordinary differential equation solvers. And there are many programs to do that. For example, you can use R, you can use Python, you can use NumMem, and several other programming software that you can use. But the problem, not really a problem, but the tricky thing here is that whoever wants to use ordinary differential equation solvers to write PBPK models, they have to have domain expertise in that particular programming language. For example, if you want to develop a PBPK model in Python, at least you must have an understanding of, you know, writing a code in Python. So that is one of the hurdles. Another hurdle is that you have to be able to understand the anatomy and physiology of the biological system that you want to develop. So you have to understand how does blood flow from the heart to the lung, and then back to the heart, to the rest of the body. So all those things, you have to be versatile, you know, you have to be vast in those areas and then be able to bring them together as an equation. So because of all this, it makes developing PBPK model with ordinary differential equation a kind of difficult task, especially for beginners. Okay, so this is something that, you know, we aspire to do at a higher level. Okay, then the second way to build a PBPK model is to use, you know, ready-made software that are user-friendly. So the interesting thing about these platforms, which I call graphical user interface platform, is that, you know, they've already taken care of all the system parameters, all the dynamics, the blood circulation, how the drug is being distributed to all the other parts of the body. So the only thing you need to do here is basically to put in the parameters of your drug, and then you simulate straight away. So because of this, it makes it very easy to build a PBPK model using these platforms. And then once you are very good with this platform, then you can work your way up to, writing PBPK model equations for yourself, from the scratch. All right. So like I said, using these GUI platforms is easy, and then it's a great starting point for beginners, because as a beginner, if you start with ordinary differential equations, you may get discouraged. So, but if you are using this kind of softwares and then you are simulating, you are seeing the profile, that can stimulate your interest, and then you want to learn more along the line. Okay. Now, there are several PBPK softwares, but the most common one, I have put them on this slide, in no particular order. So we have SimSip, which was developed by Setara. It's a graphical user interface for PBPK model. We also have PKSim by Open Systems Pharmacology. And then we have GastroPlus by Simulations Plus. So all these are softwares that you can use to build a PBPK model. However, only PKSim is an open source platform that is free to use for researchers, academicians, or people who just have interest in picking up building PBPK models. Okay, but I know maybe some of these other software also, if you are in academics and you want to use it for training or for your research, they also give academic licenses, so you just have to request for it. But for the purpose of our discussion today, I'm going to be focusing on PKSim because it doesn't require any license or academic request for license. Okay. So few weeks ago, we got instructions on how to download and install PKSim on our laptop. And so for the interest of time, I am going to be assuming, I hope I am safe to make that assumption, that all of us have downloaded the software and it's ready to go for us on our laptop. So if you have not done so, maybe you can start, open your PKSim down, and then we can walk through it together. So what I want to do right now is to demonstrate all the interface for you and describe each of the sections. And so I am going to be switching to PKSim, and then we are going to come back again to continue before we finally do our hands-on. Okay. So here is the PKSim interface. So when you are developing a PBPK project, the first thing that you have in mind, basically, I would say this, that if you want to develop a PBPK project, you always have a drug in mind. That is because you can't just develop a model of a human being and then you are not interested in doing anything with it. So as pharmacokinetics oriented people, our interest is to develop a model and then simulate concentration of our drug. So my point is that anytime you have a PBPK project, you have a drug in mind. So because we have a drug in mind, you have to understand, what is this drug being transported? What are the enzymes responsible for the metabolism and things like that? So once you have those information, the first thing you want to do is to come to this tab and then put in those enzymes, proteins that are responsible or liable for the drug. You first need to load them. So what you are doing here is that you are putting those information in the library for that particular project, so that along the line, you are going to be able to easily have access to them. So that's how you do it. So you come here, you load all your enzymes, all your proteins, PGP transporters, ABC, OATP, and several of them, okay? Whichever is applicable to your drug. Then once you do that, you will develop an individual profile. So basically here, all the system parameters are already defined for you. The only thing you need to do is to say, this individual I want to simulate for, what is the age, what is the weight, what is the BMI, and I'm going to demonstrate that later. So I'm just describing the interface right now. And then once you are done with the individual, then you can develop a population based on that individual. For example, if we specify an individual who is 20 years old, a weight of 70 kilograms, And then if you want to develop a population based on that individual, so what this population does is, okay, the average age is 20, the average weight is 70, simulate 100 individuals of this type for me. So what the population would do is it will just simulate randomly and then give you 100 or 200 or however many you specified number of subjects that you want. So, and then for each subject, all of them are going to be varying a little bit. Maybe some will be 18 years old, some might be 19 years old, some might be 22, 23, but the average is going to be based on the individual profile that you have created. And another way is you can specify maybe the age range that you want and a lot of things. So we are still going to demonstrate that. I'm just telling you what each of the tab is representing. Then once you are done with that, you want to set up your compound profile. So all those information that you have gathered about your drug, lipophilicity, maybe solubility, dissociation constants, and several of them, this is where you put in those information, as well as how is your drug metabolized. So you put in all those information here. Then for the formulation, we don't really need this if you are developing for IV model, okay? But if you are developing for an oral drug, for example, tablet, then you need to specify the formulation, okay? Then we have administration protocol. So what administration protocol means is you want to define your dosing regimen. So how often do you want to give this drug? Is it 10 milligram every day, 10 milligram twice a day, thrice a day? So here is where you specify all those things. And then these two events, as well as observers, we are not going to be using it today, and it's a little bit on the high level, but I can tell you what they do. Basically, for events, we use it in a situation where, let's say, for example, you want to test the effect of if someone is taking the drug on an empty stomach, you can specify that. If the person is taking a certain type of meal, maybe a low-fat meal, a high-fat meal, so you can specify those things, and then you simulate what the profile will look like. And then observers is basically saying you are observing maybe your concentration time profile in a particular organ that you are interested. So, so you can observe in that particular organ, and you can also connect it to another proprietary software by Open System Pharmacology, which is called MOBI. So like I said, this tool is a little bit advanced, so we are not going to be focusing on this tool today. And then you can perform simulations once you are done with specifying all this information, okay? So when you want to, let's say you want to load, or you want to start by putting information in the first block here, you can either do it from here, you click it, and then you add metabolizing enzyme, if it's an enzyme you want to add. If it is a protein, you can click it here and then you add it. If it is a protein binding partner, you can also do the same, okay? So this is where you specify. Alternatively, you can also come to the building blocks here. So if you want to do it from here, it's either you double click, okay? Sorry, you right click and then you select. You right click and then you select. Then you can select what you want, okay? So the same thing here, you can click here and then you can specify information about your individual, or you can come here and then you right click and then you do it that way. So either way is fine for the software, okay? So this is basically the interface. This is how it works. And then if you want to simulate, this is the tab where we perform simulations, okay? So I'm going to go back to the presentation slide now. All right. Now, we have looked at how the software looks like. We have a feel of what the interface is. Let us now take a case study and then work together on this one, building the PBPK model, and then we can practicalize whatever we have learned so far. Okay, so we are going to be using an anti-cancer drug, or agent, which is called imatinib. So imatinib is a tyrosine kinase inhibitor, one of the very first generation, that is used in several cancer treatments. All right. Now we know that it is given 400 milligram per day in adults. Okay. And we know that the optimal therapeutic range is between 1,000 to 3,000 nanogram per ml. Now, here is an hypothetical question that we want to answer. Okay, so mind you, I said hypothetical because this drug has been approved for a very long time. It has been extensively studied. So some of these questions may have already been answered. So, but this is just to like give you like something to work with so that we can see how to build PBPK model or understand the workflow that is involved. Okay, so in this hypothetical scenario, okay, we are assuming that imatinib has only been approved in adults. We have not, so we don't have any information in pediatrics yet. Okay, like I said, it's an hypothetical scenario. The drug has been extensively studied both in adults and children, but for the purpose of our PBPK model today, let's assume that imatinib has only been approved in adults, and then we don't have information in pediatrics. Okay, but we know that the mechanism of action of the drug, as well as the response of the tissues and cells to that drug, is similar between adults and pediatrics, because you need to establish these two conditions before FDA can accept that, okay, fine, instead of you doing another study in pediatrics, you can use a PBPK model and tell us what the dose should be in this population. Okay, so you see, our goal with this project is that I am a company, I developed this drug, and I have given you the dose in adults. I have conducted a study in adults. We spent several millions of dollars, and now we want to extend it so that pediatrics can also use it. The normal golden standard rule is that, okay, fine, you want to extend the application to pediatrics, go do a study. Okay, so but because the study is very expensive for pediatrics, then as a company we can approach the FDA that, okay, I have results from adults, I can do a PBPK simulation, and then I can use this to have an idea what the dose would be, and then you can approve it based on that. So if the company succeed with this, they are going to be saving millions of dollars. Okay, and that is something that is interesting. I'm not just saying this hypothetical now. This is something that pharmaceutical companies do, and they are able to get away with spending, a lot of money, wasting time in clinical trials. So you can actually use this kind of modeling approach to answer those questions. So here we are looking for, okay, do we need to adjust the dose in these children and adolescents? And mind you, when we say pediatrics, FDA language, pediatrics simply covers how do I put it now? People between the ages of 0 to 17. That's what FDA refers to as pediatrics. And then we can separate it out from 0 to 1 year is infants, from 1 year to 2 years is, from 0 to 1 year is neonate, and then from 0 to 1 month rather is neonate, then from 1 month to 2 years is infants, then from 2 to 12 years is children, and then from 12 to 17 years is adolescent, based on FDA guideline. Okay, and these are the demographics of these children and adolescents based on CDC growth charts. Okay, so again, what we want to do here is, this drug is approved in adults, is given 400 milligram per day. Now we want to see if I want to ask pediatrics to use it, basically children and adolescents, do we tell them to use the same 400 milligram, or we ask them to reduce the dose? If they are reducing the dose, how much should they get? So that's the question we want to answer. Okay. Now, this is the workflow that we are going to adopt here. And we are going to be starting with building and validating an IV PBPK model of imatinib, okay? And the reason why most PBPK projects starts from building an intravenous model is very simple, because if you give a drug IV, there's no absorption, there's nothing like that. So you give the drug straight into the bloodstream, and the only thing that is happening is distribution, metabolism, and elimination. So because absorption is complex for some of us who are familiar with the in that area. So, but if I give a drug IV, we can easily build a PBPK model that can tell us whether we are good with the distribution, metabolism, and elimination. Okay, so if we succeed with this, then I am happy that okay, my distribution of the drug is fine, my metabolism is fine, my elimination is fine. Then you can add on that. So when you are adding on that, let's now say we have this, we are happy with it, validate it. Then you take that model and then you incorporate absorption parameters, okay? So what we are doing in this step is basically to incorporate absorption parameters. So when we incorporate absorption parameter, then we can now say, okay, this is an oral tablet, or if it is a subcutaneous injection, or it is a capsule, however you want, okay? So incorporate absorption parameters, and then we would also look for information about the parameters that are describing this absorption, and then we build the model, we validate it as well, okay? Now, I've mentioned validate like twice now. The question now is, how do we then validate? Basically, how we validate is that I am going to look into literature. I would look for situations where imatinib has been given to adults in clinical trials. I am going to digitize the PK profile, and then I am going to compare my model simulations with the results from a clinical trial. So if my model is able to simulate what was actually done in the clinical trial for adults, then I'm happy with that. I can say the model is validated. Then I will do the same thing for oral tablets. I know the drug has been given orally, is currently being used orally. So I will take results from adults oral administration, from literature or from clinical trials that have been conducted. And then I will compare the result with my model. Then I would, fine tune the model, make sure I validate it, and then I am happy with the model. Then we can now change the system parameters from that of an adult to that of pediatric that we want, and then we can now simulate. So that's basically the 3 steps that we are going to follow. Okay, so I said we want to validate. So that means we need some clinical trials where this drug has been given intravenously, and we need some clinical trials where this drug has been given orally. So that is the first thing you want to do. I want to check literature. Are there places where clinical trials have been done? Do they report the PK profile? So if they do, that is what I'm going to use. Okay, so I have kind of done that for us. Okay, so in order not to waste time and save us a lot of time, so I can see Peng et al. In 2004, they actually had a clinical trial where they gave 100 milligram of imatinib intravenous infusion to healthy volunteers. And then this particular profile is the one belonging to the IV infusion. So that's the only one that we need, this particular one. So we need that. And then in 2004 as well, Peng et al. Also did another study where they studied several formulations, but I think I cut off some of the information. But this particular one in the middle is the one that is 400 milligram tablets, the one in the middle here. So you get that information. Then there's also another study by Nikolova et al. In 2004 that was given 400 milligram of the tablets. You see the one in square. Okay, so, and this is what the profile looks like. So I have all this profile. and then you are going to digitize it. I want to believe most of us are scientists, so we know how to digitize. Basically, what digitize means is that I'm going to use a software, and I'm going to be using it to pick all these points and then convert it into concentration, and then we can use that for simulation. Okay, so let me quickly show you how to do that in case of those who are just being exposed to something like that. So I'm going to drag my screen here, and then you can just say plot digitizer. So it takes you to this interface, and then you can upload so you are going to get the image of that profile. You can screen grab it and save it, and then you go to that image. I have it already, so I'm going to just use one of them. So it's going to look something like this, and then it's going to say, where does your X1 start from? You click and drag. I put it here. Where's your X2? I'm going to put it at the last time here. So X1, I put it at 0 point. X2, I put it at 100. And then I'm going to put Y1 at the beginning here. I'm going to put Y2 at the end. So Y1 is at the origin, which is 0. Y2 is at 1,800. Okay, so once you have that, then you can now start to click the point that you want. So in this case, we wanted 400 milligram tablets because we are interested in tablets. So this is the square here. That's the tablet. So then we can start click, 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 and then you keep going. Make sure it's almost perfect. So once you do all that, you are going to be able to then copy these results and then save it in an Excel file. Okay, so that's the reason why you are digitizing it. So you can copy it and then you can use it where you want to use it. Okay, so that is how to extract those information from the data. Okay, so once you are done with that, then the next step is, like I said, every PBPK models, you need system parameters, you need drug parameters, and you need dynamic equation to describe the flow of drug all over the body. Okay, so right now with PKSIM, what we have here is that the system parameters has already been defined. We don't have to worry about that. The fluid dynamics, the blood flow to all over the body has already been provided, so we don't have to worry about that. But you need information about your drug. PKSIM does not provide that, so you have to look for it and then provide it, okay? So for the interest of time, I have also looked for this so that it doesn't, the webinar doesn't take too long. So I've done the research. So here are the information that I found. So the physicochemical properties, the solubility, the pKa, the log P, the molecular weight, effective permeability, etc., etc. So these are the information that I am going to be using to build the PBPK model. Okay, so I'm going to pause a moment here for like 2 minutes, and maybe we can digest that, and then we'll go back to the interface and answer the question. Okay, so we can have like 2 minutes and then we'll continue shortly afterwards. Okay, so we are back, but I wanted to quickly mention here that if for example, if you look at the fraction unbound, the fraction unbound of imatinib is between 2.94 to 5% because different sources report, some differences. So if there are some certain things that are like very constant, for example, the molecular weight of imatinib is 493.6. The instrumentation that determined that is highly accurate. So there's nothing you can do about this. It's probably very correct. But there are some values that, yeah, it is being reported, but if you check 2 or 3 literature sources, some, for example, for lipophilicity, some can say it is 3, some can say it is 3.5, some can say it is 3.4. So in those situations, you have wiggle room so that if my profile is not looking very good, I can adjust those parameters because, literature sources are not really giving me certainty on what the exact value should be. So I'm preparing your mind now that, some of these parameters, if you see that your profile is not looking 100%, you can walk around it and maybe do some optimization. So we call it optimization. Okay, so, or if you like, tweaking. Okay, so I'm going to switch now to the software back. All right. So let me drag this to my other screen so that I can see the parameters and use it. Okay, so the first thing we want to do is to build up our expression profile. And like I said, from what I showed you earlier here, the drug is being metabolized by CYP3A4, CYP2C8. So those two enzymes, take note, CYP3A4, CYP2C8. Okay, so since we know that, then I need to build an expression profile. Okay, so I want to add metabolizing enzyme. The first one I want to add, so you can, this metabolizing enzyme, just click the drop down. You can search CYP3A4, then you select. The phenotype is for elderly individuals. So if you are working with maybe another population, you can select it here, hepatic impairment, or what have you. So, but we are dealing with healthy subjects, okay? So you just click OK. And so again, I am loading those enzymes into my workspace so that I can use them later, okay? So that's the first enzyme that is liable for our drug. The second enzyme is CYP2C8. So CYP2C8, can look for that as well, and then select it. Okay, now that we have the two enzymes selected, okay, we'll go to the next thing, which is we want to develop individual profile. Okay, so let's develop individual profile now. So you click it, and then what do we want to call this individual? So let's say this is healthy volunteer, healthy volunteers. Okay, and then let's say it is a white American. So you can either pick a male or female gender. So if you look at the table again, for some of you that screenshotted it or something like that, I screenshot it rather. So you would see that it's basically 82% male in those healthy volunteers that participated in that clinical trial. Okay, so what is the average age that we want to use? So from that thing that I showed, I think it was around 38. And then, so if I click a number here, 42, you see all those other parameters populate automatically. So there's an equation under the hood that is doing that. Okay, but if you have the exact value from the clinical trial, you can replace it as you want. So in this case, I want to say 38. And then the weight is about 77. Okay, 77. And then do we have information about the height? The height is 177 from the clinical trial. And then they didn't give us information about BMI, but of course, if you know the weight and height, you can get what BMI should be. All right. So now that we are done with that, then we can click next. If you scroll down, this is not showing my next, by the way. I think the resolution is too big.
If you double click where it says create individual, just double click the top part. There you go.
Okay. So next. So here is basically telling you the parameters that were in the model. So like I said, you see the bone, the brain, the fat, what are the value for the organ weight, everything has been specified. So you don't need to really change anything here. Things describing physiology, the flow rate, the glomerular filtration, all those has already been defined. So you don't need to do anything there. And that's the beauty of using this graphical user interface, okay? So we just say next. So here we have to specify. So that's why I said you need to initially load the metabolizing enzyme. So here I have to tell it that for my individual, this healthy volunteer, I want you to metabolize the drug with CYP3A4. So I'm adding it for the individual. And then I also want to add CYP2C8 for the individual, okay? So what that means is that any drug that I am giving this healthy volunteer, that drug is going to be metabolized by these two enzymes. So that's what that means. If you don't initially specify it in the expression profile, then it's going to automatically create it for you. But it's good practice to start from, the way we started it, okay? So then you just click OK, and then we are done with the individual, right? Then for the next part is the population. So if we click the population, we want to name our population as well. So let's call it healthy volunteers population. Or let me see, volunteer pop. Okay. So then it's going to ask you, is it based on, so if you have more than one individual developed in the previous step, then you have to select which one you want to base your individual on, your population on, okay? So we want to base it on the healthy population. How many subjects do you want? Let's say we want 100, let's keep it 100. And then what is the proportion of females? So depending on what you want, I want to put 16 here, because in the clinical trial, I think it's about 82%. If you look at it again here, you see the population is 82% male in those clinical trial. So I want to use values that are as close as possible to what was reported in those clinical trial, okay? So now this is 16%. Then what is the weight range? The age range. So in the clinical trial, the age range is between 19 years old to the height. The height is between 161 centimeter to 194 centimeter, and then the BMI is going to be automatically calculated. So once you specify all this from the clinical trial or the publication that you are using, then you click next, and it's going to create the individuals for you. So here is the metabolizing enzyme. You don't need to specify it here because the population is based on that individual. So since we already specified those enzymes for the individual, then it automatically carries it forward, okay? Then again, we don't need to change anything here. It's already specified, and we can just click next, and then okay. So we have our healthy volunteer, we have our healthy population, okay? Then the next step is to have our compound profile, So you can right click here and then add compound, or you click it here. So either way it works. All right. So what is the name of our drug? It is imatinib. And then time is going very fast, so I'm going to be a little bit faster now. So what is the lipophilicity of imatinib? So it is about 3.5. Again, like I said, this is not set in stone, right? Because you can come back and optimize it. Because we are not really sure of that number, there are several sources that are reporting different things for the log P. So, but this is what I'm going with for now, for a start, okay? Then the protein binding, does the drug bind to albumin or it binds to alpha-1 acid glycoprotein? Okay, so I also showed it that it binds to alpha-1 acid glycoprotein, and then the fraction unbound, let's start with 0.05, that is 5%. Okay, but if you want, you can also specify in percent, and then you just say 5, whichever one is fine. Okay, so can either specify in percentage or you specify the actual value. So I like to use the fraction. All right. Then if you in a standard PKSIM work profile, you have to specify where you are getting those information. So for example, this log P, let's say I got it from PubMed. So I'm going to say here, database, what is the name of the database? I will say PubChem, and then you click OK. So it's going to tell you value, where is the origin for this value you are using. So you say PubChem. So you can put the value. So if it is a paper, let's say I'm getting this from a paper, so I will just say publication. What is the name of the publication? Let's just use Pharmacometrics Africa 2025. So you can basically name each of those things accordingly, okay? But obviously, I'll just remove this since this is not true. But let's just leave it like that, okay? Now, you specify the molecular weight. The molecular weight is 493.6, okay? Does it have halogen? No. If you check the the molecular structure, there is no halogen attached to imatinib. Then what is the compound type? So again, if you also check online, you will see that imatinib is dibasic in nature, and one of them is like 8.07. I think I showed this earlier. I don't want to keep going back and forth between the PowerPoint and here. And then 3.03 for the second base. Then what is the solubility? You have to specify 14.6. The unit has to be consistent. You have to make sure. If the unit is something else, you have to make sure you adjust it appropriately, okay? So then now we are done. Let's, let me see. Okay. So now that we are done, then we click next, okay? So in this section, you now have to say, for my drug, how is it going to be metabolized? So you need to specify, I want to add a metabolizing enzyme. What is the enzyme you want to add? The primary one is CYP3A4. Where did you get information about this enzyme from? Then you can mention it. So I'll just say paper, then paper 1, let's say paper 1. What is the metabolite? NDMI. So so if let me just show you very briefly. I just want to show you that you could actually get this information online. All you just need is to search. So as you can see here, the ionization, it is diprotic base, the PK 8.07, 3.73, the blood to plasma ratio, the fraction unbound. So protein binding component, alpha-1 acylglycoprotein. So all you just need is to search. You will see all those information about that particular drug, and then you can use it. Okay, so everything I'm doing here is based on what I have searched. So if you are doing your own personal project, all you just need is to look for those values, and then I am very sure you are going to find them. Okay, so for CYP3A4, I'm going to select what the value is. So the paper reported a Vmax of 3, and then the unit is picomole per minute, picomole per minute per picomole of enzyme, picomole, yeah, this one. Then the Km is 10.54, okay, and then micromole per liter, okay. Then the paper also reported that there's another metabolite from that particular enzyme, so they call it others. So let's say paper 2, the name of the metabolite is others, and then they reported it as there's a 3.4 microliter per minute per milligram. Then okay, so 3.4, and then if I click OK, so that is for CYP3A4. Those are the two enzymes. Then we also have for CYP2C8. So CYP2C8, where's the data source? I'll just say paper 1 and paper 2 for the sake of time. NMDI. Per minute per milligram of protein. So this is correct. And then the Km is 7.49, and the unit is micromole per liter, micromole per liter. So this is good. And then the last enzyme the last metabolism that we want to add is, the paper also said the drug is also metabolized into other metabolites, and then we want to specify that. Okay, so the value is 24.2 microliter per minute, from protein. Okay, so then I click OK. So we have specified all the potential mechanism by which the drug is being metabolized. All right. Now the paper also mentioned that there's also some contribution from renal, very minor. Okay, so I'll just say paper and then the plasma clearance. So the paper reported liter per hour per kilogram. So if I bring up the paper for a minute, just to show you that I'm using what has been reported. So if you look at here, they say renal clearance is 0.5 liter per hour. So that is what I am using in this case, okay? But if you take note here, it is liter per hour per kilogram. So I'll just divide that number by 70 and then get an idea for the ballpark of what that value should be. Okay, so 0.5 divided by 70, that would give us 0.00714. Okay, so by the way, you don't have to do this unless the information about your drug is suggesting that it is being metabolized by both hepatic pathway as well as renal pathway. Then you incorporate those information. So if your drug is only metabolized renally, then you neglect the part of the CYP enzyme. If your drug is being metabolized only by hepatic mechanism, then you don't need to specify anything under the renal clearance. So, but because imatinib has kind of both components, so that's why I'm filling for those two parts. Okay, so I think after that we are done. Basically, you don't need to adjust anything here. So then we click OK, and then our compound profile is set up. Okay. Then what we want to do next is under formulation. This is only for non-IV route, so I'm going to leave it for now. So I'll just go to administration protocol, and then I would say, let's say we want to give 100 milligram IV, okay? And then it's an intravenous infusion, 100 milligram, the dose is milligram, and then it's given every 24 hours, okay? So once I specify that, I'll just click OK. And then I have specified the dosing protocol. Give 100 milligram at time 0. That's it. If I want it to repeat, I can say 48, and then it's going to give 1 at time 0, and then it's going to give another dose at 24 hours. So, and then everything ends at 48. Okay. So, but I'm going to, for simplicity's sake, just use 24 hours for now. All right. So we have expression profile, individual, we have the population setup, we have the compound, we have the administration protocol. So let us simulate. Okay. So we add simulation. So you have to name your simulation as well. So let's say 100 milligram IV IV simulation, and then okay, so you click next, and then so here is also another part that you have to really pay attention to, because you have to do some optimization here. What I mean is that there are several methods that you can use. I think I described this when we were going through the lecture, that there are several ways you can calculate partition coefficients. So you can use Rogers and Roland, Schmitz, Pauling et al. And all. So you have to try them one by one to see which one is the best, and I will show you how. But for now, let's just go with the first one. What we see here, I'll just click next. And then how is the drug metabolized? Renal clearance, and then we have also hepatic clearance by the enzyme. But here, I want to add the one that has the other mechanism, and then I'm going to add the other one for the other mechanism. Okay, so next. So here is the dosing protocol. I only have one, so that's why it's only showing one. Okay, so then next, if you wanted to check for food effect or things like that, or a particular observation that you want to observe in any organ, you can also specify that. But like I said, we don't need these two for now, and the program is still gonna run. Okay, so I'll click next and then okay. So it has created the simulation profile for us. Is going to look like this. And then you are going to come here and say, now that I have loaded the simulation, then I need to run it. So then you run it, you click run. So when you click run, it's going to ask you this basic question: where do you want to simulate? Is it in the plasma? Is it blood? So you can change it as you want, but most of the time we simulate plasma, and in clinical trials, plasma is what is measured most of the time. So for this drug, in this case, it is plasma, so we leave it like that. I'll just click OK. And here is our simulation results, okay? Now, if you look at so we have a profile now, but if you look at the units of if you look at the profile that I showed earlier, if you look at the units, the unit is in nanogram per ml. Nanogram per ml. Okay. So because of that, I want to also work with nanogram per ml. Okay. So if you look at the units here, it's in micromole per liter. So if you want to change the unit, just double click, and then you change the unit. Very easy. It's just click, 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 and then you get that done. So here is the nanogram per ml. But the question now is that, how do we know if this simulation is even correct at all? So what we have done up to now is we have built a PBPK model, but the next step we are now going into is I want to validate, I want to check if my model simulation is correct at all. So for you to do this, then you have to add observed data. So that is where that thing that we digitized, the profile that we digitized, that's where it comes in. So you come here, you right click the observer, then you add observer data. I have already saved it because I've done it before. I've digitized it earlier. So this is the IV 100 milligram. Okay. So you first have to specify what is the unit of time in that paper. It is in hours. What is the unit of amount, concentration? It is mass concentration. So you select mass concentration, and then you select the appropriate unit, which is nanogram per ml. And then, okay, so that's done. Then you have to click which species was that study done in. So that study was done in humans. Which organ was the concentration collected? Peripheral venous blood, and then it is plasma. So what molecule is the drug? It is imatinib. So you select all this, and then you are good to go. Then you say, once you are done with that, and then if this is what I want to copy, I will go here. If it is this one, if it is this, so but I just wanted the IV for now, and then add current sheet. Okay, so you can rename it whatever you want, but I'll just leave it as it is now for the interest of time. And then you just say import. Okay, so you say I now have that profile already in my workspace. So if you want to bring the profile to your simulation area so that you can compare, then you can click this and drag it. Okay, so now that I have dragged it, we want to say, is the profile matching? Is it not matching? I think this name is too long, so it's covering the CMAX. So I want to rename it to IV 100 milligram observed. Okay, so I'm going to remove it here and then I'll drag it back. Okay, good. Now you can see that we are off from the CMAX. So then you can start to try several different things. Okay, so if you want to edit, you just come back to configure, and then you can say, now instead of using PKSIM standard, what if I use Roger and Roland? How does the profile look? So you start to check, you do basically try and error. So if I use this method instead of the previous one, what happens? Then you run it again. Okay, so you see this is what happens. Then you keep trying different things, and then different combination, Rogers here, this one there. But for the sake of time, I've tried it and I know what works, so I'm just going to go there. If you are practicing on your own, you can do that, and then probably you'll find whichever works for your drug. Okay, so but you are definitely going to find one. So I'm going to stick with Rogers and Roland, and then use cellular permeability of charge-dependent Schmidt. So if I click OK, okay, so then you now simulate. You run it again. You run it again for you to see the changes. All right. So it's too big. Okay. So here is what we have right now. But if you look at it, I am still a little bit off from the CMAX. It's a little bit off. Again, you can't be 100% perfect, but I want to achieve as close as possible. So it's possible, but you can't get 100% perfection. That is for sure. But you can try. Okay. So based on what I have done, I'm going to go back to the drug compound. I want to bring the drug back. If you want to bring the drug back, you just go to the compound profile and then you double click it. Then it's going to bring it up and then you can change whatever you want to change. Okay, so what I want to do here, I have optimized this before. So in your own private time, you can also optimize as many as you want. So I want to optimize this to 3.4 because I think that's what works the best. Now, the moment I change anything about the compound, you can see that in the simulation tab, it's telling me exclamation mark, something has changed. So what you then need to do is to right click here and tell it that, okay, I have changed something. I want you to update it from the block. Okay. So then you say update and then it automatically updates. So once you do that, you go back to the simulation and then you simulate again. Okay. So this looks a lot better for me. All right. Then this is for just one individual, if you remember, just for one individual. Okay. So if you want to simulate for a population, so you just say add simulation, again, 100 milligram IV population. Okay. So now population is what we are dealing with. Okay. So instead of picking healthy volunteer, you are going to pick population, healthy volunteer population that you initially created. And if you hover over it, you will see the parameters of the population like the age, the weight, the height, the BMI. Okay. Then you click next. So if you remember, we used Rogers and Roland, and then we used child-dependent Smith. You click next. You have to select the second CYP3A4, then select the second CYP2C8, and then next, 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 and then okay. So it's going to create a population simulation. Okay, so now we have this. It has loaded the simulation for the population. Then you click run. Again, it's going to ask you where you want to simulate. So plasma, the same. You click OK. And then it's going to ask you a couple of questions afterwards. All right, so now it's asking you here, I'm going to give you arithmetic mean. What kind of variation do you want? Do you want standard deviation, or you want 5 to 95%, or you want geometric standard deviation? So whatever you want, you can pick. Okay, so basically you pick one central measure of tendency, and then you pick another one that is like a variability. So, but typically we usually use like arithmetic mean or geometric mean, and then you specify 90% prediction interval, which is basically this one from 5 to 95%. Okay. So then my units, I want it to be nanogram per ml. I want it to be linear scale. Okay. You can choose whatever you want, but you have to be consistent with your observed data. So then next, you click next, next, and then the profile is already here, but you click OK to actually have the simulation. Okay, so as you can see here, for this one, it's just for one individual. That is why you're only seeing one profile. But if you do it for a population, then you are going to see like variability from one person to another person. Okay, so again, you can also check your observed. You just click it and drag it to this space, and then you can see how the profile fits. So you can see here, it looks like we have really done a very good job. So up until now, we have now developed an IV model. We have developed an IV model. And remember what we said? We said for the IV model, we are good with the metabolism, we are good with the elimination, and then we are also good with the distribution. So that means because I am fine, I believe I have, validated this model, then I don't need to touch anything about distribution. I don't need to touch anything about metabolism. I don't need to touch anything about elimination anymore. So then we want to go to the next part, which is then to have our, so here we said, here is it again, the workflow. So we said we are going to build adult intravenous model. We have done that. So now that we have done that, then we want to move ahead to building for oral tablets. So I'm going to do that very fast because of time. Okay, so I'll collapse this, and then so for the oral tablets, I already have the healthy volunteer. I don't need to repeat that. I can use the same individual. So I'll just clean the space. I'm trying to clean it so that I have a clean space. Okay, so what I need to do now is I have, okay, so the individual is the same. The population is the same. Nothing changes about that. The drug is the same, so I don't need to change that. But what I need to change, in fact, is the administration protocol. So right click on the administration protocol, then say add administration protocol. I want to call this 400 milligram tablets, because that's what it is. Then here, instead of IV, you would select oral, okay? And then what is the dose? The dose is 400 milligram. What is the unit? The unit is in milligram. And then given every 24 hours, okay? So then you click OK, and that's it, okay? So we now have for tablets, all right? Okay, tablets, we have it. Then let's now simulate for the tablet and see what the profile will look like. Let's simulate for the tablet, okay? So I'm going to create a simulation for the tablet. I'll call it 100 milligram tablet simulation, okay? So I want to use the volunteer because I only want to start with one individual. Then when I'm good with that, I can simulate the population, okay? So you click next. Again, so you have to make sure that, because like that's what I said about the initial step, that whatever you have established for the IV model, that is what you have to continue, because all these things are what describes the distribution, the metabolism, the elimination. So at the point of adding oral route, you are not going to change the metabolism, because if you give a drug orally or by IV, the metabolism doesn't change. The enzyme that will metabolize it will not change. If the drug is liable for renal clearance, that is not going to change. So all those things will remain the same. You don't have to start another new trial for this part. So anything you want to concentrate at this point is on the absorption parameters, and that's what you should focus on. So I'm going to leave it to what we had earlier, okay? Then for the processes, again, I want to use all the processes for the SIP. One is NMDI metabolites, the other one is order. So I want to make sure everything is specified. Then administration protocol. So now we want to do for tablets. So you have to make sure that you change from IV to tablet. You change from IV to tablets, okay? So I'm going to select tablets. Now, when you select tablets, it's going to ask you for an extra information that what is your formulation, and that is where the formulation comes in. So then you have to create a formulation. So how to create a formulation is come to this plus sign, and then you click that. So because of time, again, you can explore as you want on your own, but I'm going to explore a weighable formulation. Okay, that's the most common one that you can easily optimize. Okay, so I'm going to use a weighable formulation absorption model, and then we'll see how that performs. Okay, then next, okay. Okay, so this is our 100 milligram tablets individual simulation. Then I'm going to simulate now. Okay, and simulate. All right, so this is my micromole per ml. I want to change it to nanogram per ml, and I want it to be linear. So okay, so we have this. Now we don't know if this is correct or not correct, or things like that. So how do we know? We have to get what has been done in clinical trial, and then we want to match it. Okay, so then I'll go back to my observed, because I can't put the IV. This is oral. So I'll go back to my observed, and I'm going to add observed data for oral. So just the same process. You do the same thing, concentration, mass concentration, nanogram per ml, species, human being, organ is peripheral, plasma, imatinib, then 400 milligram by Nikolova, add it. I want to rename it 400 milligram Nikolova. Okay, and then I'm going to import. So if you come here, I've already imported it, so you can click it and drag it to the interface. Okay, then I think I have two observed data for the tablet, so I'm going to quickly load the second one as well, and then we can walk through how to optimize the parameters. So concentration, mass concentration, nanogram per ml, species, human being, organ is peripheral, plasma, imatinib. And then I want to add this second tablet. So you go to that and then you add it. And that is 400 milligram tablet. This is from Peng et al. Just to differentiate between the two. Okay. So I want to rename this so that it can be consistent. Okay. So what I have currently is the one from Nikolova. So I want to add the one from Peng as well. So you see, now we want to match this profile. Okay. So what do we do? Again, like I said, don't touch distribution, don't touch metabolism, don't touch elimination, because we have already done those for the IV. So you don't touch it. If you touch those parameters, then that means you have already affected whatever simulation you have done for IV. So that's why we don't touch it. So the only thing you can optimize, or you can play around with here, are things that has to do with the absorption. Okay, so let's start. The first thing that you can do is let's go to the Weibull. So the dissolution time is 240. If I reduce it to 120, and then you can see what happens. Okay, so anytime you change anything, you have to commit it, and then you resimulate. Okay, so the profile went up a bit, but it's not responding very well. I'm just going to give you what I did to make it work, and then at your own leisure time, you can try as many things as possible. So what I did was I go back to the compound. I went back to the compound. So basically, things that are affecting absorption is going to be here, absorption. Can you see here? Okay, and then we have specific intestinal permeability. Now, currently, the software is calculating it for us using some equations that I have described in our earlier talks. So there are several equations to predict this. But then what I did was I wanted to use what this paper reported. So if you look at this paper, where is it? Effective permeability was reported as 0.92 exponential minus 4 centimeter per second. So that is what I want to use as for the optimization. Okay, so I'm going to come here, add, and then I'll say P effective permeability optimized. Okay, So instead of using the calculated one, I want to put a value that I want. So that's 0.92 exponential minus 4. Okay, the unit, again, if you remember, the unit is centimeter per second. Okay, so centimeter per second. Okay, now I'll go back to the simulation. I'll go to configure, and then I can go to compounds. So you see here, these two you don't change because they affect distribution. Okay, but here is something that affects absorption. Okay, so instead of using the calculated value, I want to use the value that I just optimized. So I will click that, and then I will click OK and say how the model performs. So I'll click run, and then that's it. It's a lot better. Okay. Now, since it's a lot better now, what else can I do to help this? I went back to the Weibull. I'll just tell you what I did because of time, so I don't want to talk you through it. So you just, I basically used 100 minutes for the 50% dissolution time, and then I use the dissolution shape as 1. And then if you try the simulation again with this, then I think we should be good. Okay, so here it's all good. Okay, then I want to try it for the population and see if it's the same. So I'm having 400 milligram tablets population, and next I want to use the optimized one. Next, I want to select all the metabolizing metabolites formed. Next, I want to use tablets, and then I want to use the Weibull. If you have another one, you can pick from which one you want to select. Okay. So, okay. All right, then you can now say run, and then you click OK, and then you simulate. Oh, this I didn't select, so I didn't select population here. That was the problem. 400 milligram tablets, population. Okay. So next, I'm going to use Rogers Roland, Charge Smith, effective. Then add this, add this. Okay, next. Next, tablets, Weibull, next, next, okay. So then I'm going to run it. So mean and 5% to 95. Next, next, next. And then, oh, I want to change the units. Linear nanogram per ml. Next, 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 and then okay. So here is the profile. And then I want to put the observed so that I can see how the profile is doing. Put this one, and then put this one. Okay, so it looks like the simulation looks very good. Okay, so at this point, we have successfully developed the model. We have built the IV, then we have built the oral tablet. And then we can now go ahead and answer our question. But because of time, I'll just tell you what to do, and then you can probably answer that question yourself when you do that, okay? So right now, what we have is that, you know, all the simulations we have done is based on adult population. So in order for us to answer that question, all you just need to do is, okay, I'm fine with the model now. Everything looks good. So let me just delete all this. Close all this to have a clean space. So we are fine with the model, but now I want to simulate a new population. And then this new population are pediatric population. So all you just need to do is to come here and say, okay, add a new population, but this time my population is pediatric. But we have two categories of pediatric. We have the children and the adolescents. So let's start with the children. And then you can say, okay, I want 12% of them to be female. What is the age? What is the weight? Again, if you remember, we already said what those values should be here. So if we quickly, let's see, I have 3 minutes more. So this is 12. This is 40. From 2 to 12 years. 2 to 12 years. The weight is from 12 to 40. And then the height is from 85 to 150. And then for children, what is the BMI? The BMI is from 15 to 18. Okay, so let's next with that. So the CYP3A4, CYP2C8, next, and then okay. So I have the pediatric population already. So I just want to bring the simulation here so that you can see the one from adults. So you see for the adults, the Cmax is around 1,800, let's say. So let's now simulate for the pediatrics and see if the concentration is too much or too low. And then we can now decide, do I need to reduce the dose or do I need to increase the dose? So if we add simulation, I want to add, let's start with 400 milligram for the children population. Okay, so children population, I want to do 400 milligram for them. Next, Rogers and Roland, Charge Smith, effective permeability optimized. This one, this paper, the second paper. Next, we are given tablets. It is viable, and then next. Okay. So if we run the simulation, I'm just showing this, and then you can do for adolescents yourself. So let's linear nanogram per ml, and then next, and then let's click OK. So if you look at the two, this is for the adults, okay? You can see that the maximum concentration for the adults is between 1.5 and 1.8. So let's say roughly 1.7. But if you look at the simulation for the pediatric population, the Cmax was around between 4,000 and 5,000. And if you remember, we said the effective concentration is between 1,000 to 3,000. So which means any concentration above 3,000 is going to have some side effects. So that means if we give 400 milligrams to the pediatrics, this is going to go beyond the 3,000. In fact, even more than what the adults are exposed to. And so you can then say, okay, instead of 400 milligrams, let me reduce it to 200. So all you just need to do is come to this administration protocol, double click it, change it from 400 to 100 milligram, simulate again, and then you keep trying it until you get to the final dose that is going to work. So I apologize for rushing at the end. It's because it's a lot to cover, but I believe you would be able to solve what the dose would be. It was my interest to actually walk you through what the final dose would be. And then, for example, if we arrive at 200 milligram, then we can say, okay, for children between 2 to 12 years, let us give them 2 tablets of 100 milligram imatinib. If it is 300, then we can say, let's give them 3 tablets of 100 milligram imatinib and things like that. So that was my goal. And that would be the end of my presentation. And the next webinar will be for population pharmacokinetics. If you have any question, I'll be happy to answer them. Thank you.
Yeah, thank you so much, Babajide. You are really giving us a lot to digest. So thank you for the concise and very loaded practical-based session on building and implementing pharmacokinetic model. While the presentation was ongoing, a couple of questions came in. While we've been able to answer some of the questions, because of time constraints, we'll harvest the remaining questions and the faculty members will answer those questions. So I want to implore participants to watch out for the recording of this video, of this presentation. Thank you. Then before we close this session, there are quite a number of important information that I would like to pass across. The first is that this particular webinar is the final on physiologically based pharmacokinetic model. So from our next webinar in June, we'll be talking about population pharmacokinetics. And tentatively, the date of the next webinar will be 25th of June, 2025. So we implore participants to watch out for any change in the dates of the next webinar. Also, I would like to also inform participants that the recording from previous webinars are available on the PMXAfrica website as well as the Nigerian webpage. And we encourage participants to visit the website for the recording and also the recording of this particular webinar. Also, there are quite a number of members that are showing interest in knowing more about PMXAfrica, what we do in PMXAfrica. We encourage them to also check the PMXAfrica website for more details about what we do in PMXAfrica. Finally, for those interested, during the course of the lecture, there are quite a number of participants that showed interest in joining PMXAfrica Nigerian chapter. For this categories of participants, we encourage them to also visit the PMXAfrica Nigerian webpage. In that webpage, there are quite a number of contacts they can actually reach out to join PMXAfrica Nigeria. So on this note, we'll round up this session for this month, and we hope that we also have the opportunity to reconnect next month for the next webinar. Thank you. I don't know if we have to add. Yeah.
Yes, let me quickly add this. So sorry for the time that we extra minutes that we took. It was supposed to be for 1.5 hours, but because we could not adequately answer some of the questions, I've sent a Nigerian number where you can send a message to join the. I can't immediately create the WhatsApp page now. So send a message. Then we will continue this conversation on. I will send you a link to a WhatsApp page that will be created for participants of the of this webinar. So please send a message to that number and I will send the link once it's created. We can then ask all our questions from on that page. We apologize again for taking extra time that we took, and we can see you next month for the next webinar. Thank you.